Hello, everyone. Thanks for being here. My name is Chauncey Fonseneau. Today, we'll be talking about forensic identification, specifically how the legal system tries to connect invisible, accidental traces left at a scene to a specific individual. We've all watched crime shows where a computer flashes a green light and instantly solves the case. In reality, forensics is actually a mix of hard science and human psychology. Today, we're going to look at how we turn daily random contacts into courtroom proof. To understand why this matters, we have to look at a basic rule of simply existing. This is that every interaction leaves a mark. It's an invisible signature. Every time that you grab a door handle or your phone or type on a keyboard, you drop biological and physical clues. Forensic identification is the process of catching those clues and translating them into facts. When the system gets it right, it secures justice, but when the human element fails, it carries a real risk of wrongful conviction. So here's our roadmap for today. It's going to be broken down into three pillars, with the first being fingerprint analysis, which is just moving past basic patterns to look at the testing protocols that experts use. Then we're going to move into forensic odontology, which is how dental data is mapped and tracked. And then we'll get into touch DNA analysis, which is how a tiny invisible cluster of skin cells get turned into a full genetic profile. We'll start with our first pillar, and it's the most classic tool in the handbook, and it's fingerprint analysis. The biology here in fingerprint analysis relies on something called friction ridges. These are the tiny raised patterns on your fingerprints. They form right before you're born and they stay exactly the same throughout your entire life. Because of the natural oils and sweat on your skin, whenever we touch something, we leave behind a stamp of those ridges. They're usually loops, whorls, or arches. In forensics, we call these latent prints, which means that they're hidden from the naked eye until we dust or chemically treat them. So, how do we go from a faint smudge to a definitive match? Analysts follow a four-step pipeline, which is called the ACE-V protocol. A stands for analysis. This checks if the crime scene print is clear enough to use. C stands for comparison, which is putting it side by side with the suspect's print to look at the tiny details. E stands for evaluation, and this is making the call if it's a match, an exclusion, or inconclusive. And then we finally have V standing for verification. This is where a second examiner runs the entire process again, completely blind. And this is the human double check. This is what keeps the conclusion objective. When we look at how this applies in the real world, we have to talk about the human factor in this process. Even though ridge patterns are unique biology, identifying them is a human process. And this is highlighted by a study by Moringa and Maluadazi in their 2026 study. Their case data showed that things like evidence mismanagement or a lack of specialized training are the primary causes of failure when trying to secure a conviction. Examiners are rarely handed perfect prints. They're interpreting incomplete, smudged data under intense pressure. The strength of the evidence lies entirely in the precision of the investigator, not truly just the science itself. With this in mind, this brings us to a core psychological framework known as signal detection theory. Because fingerprint analysis relies on human judgment under pressure, the examiner's brain is constantly trying to separate a true forensic signal from messy background noise. You can think about it as a professional wrestling like WWE. When you're a kid and you're watching this, you think it's 100% real and unscripted, just athletic competition. But then you grow up and realize there's an entire scripted storyline happening behind the scenes. Forensic analysis can be looked at in the same way. We want to believe that it's pure real science, but cognitive and contextual biases act like a script behind the scenes. If you look at it in this way, it shifts in judgment, change real world outcomes. A hit correctly catches a criminal, and a correct rejection clears an innocent person. But if an examiner is fed outside case details, like being told a suspect had already confessed, it subconsciously lowers their threshold, and this bias can actually trick them into declaring a match on a bad print. This is known as a false alarm and a wrongful conviction. On the other hand, if their threshold is too high, they can commit a miss, failing to identify a true match and letting a perpetrator walk free. So we're going to pivot now from skin ridges to something more durable, and that is human teeth. And this brings us to forensic odontology, our second technique. Forensic dentistry works because teeth are very tough. They're actually the hardest, most resilient substances in our body. Soft tissues decay, and blood and skin DNA samples can actually break down under extreme conditions. But teeth, on the other hand, can survive intense fires, severe trauma, and advanced decomposition. And because everyone has totally unique combination of alignments, gap sizes, and fillings, your mouth acts as an unalterable biological record of who you are. So the workflow to identify remains through dentistry is very meticulous. It begins with post-mortem profiling, and this is where the deceased are examined and their mouth is mapped out using photos and digital x-rays to code every single tooth. After this, investigators track down the missing person's past dental clinics to gather their ante-mortem data. And after this, the specialist overlays both sets of data onto a standardized chart called an odontogram. And on this, they check every filling shape and root angle. And if the two charts match perfectly with zero unexplainable differences, then you have a positive identification. So when does this tool become the primary line of defense? As documented in the articles by Miller in 2024 and Wood and Gardner in 2023, we rely heavily on dental identification during disaster victim identification, or DVI abbreviated. In major aviation accidents, explosions, or sudden natural disasters, traditional physical features can be completely obliterated, and standard DNA sources are often compromised. In these intense mass casualty scenarios, forensic dental records step in as the best and primary method to restore names to remains. To expand on that DVI framework, why is this method globally recognized as a gold standard? First, even when a disaster causes severe biological degradation that breaks down raw DNA strands, dental structures retain 100% identification accuracy. And because of this reliability, forensic experts emphasize that establishing international standardization for dental charts is vital for global justice and safety coordination. To build on this, on an operational level, dental identification serves as a rapid, highly cost-effective partner to modern laboratory DNA sequencing and allows investigators to clear mass casualty files efficiently without sacrificing scientific precision. However, drawing directly from the research of Manica and Gorza in 2019, we must draw a hard, bright line between two completely different applications within this field. On one end, you have the highly objective, universally trusted practice of identifying victims through stable pre-death dental records. But on the other end, we have bite mark pattern analysis on human skin. This practice is deeply controversial and increasingly viewed as junk science in the 21st century. This is mainly due to human skin being elastic. It stretches, swells, and heals and makes it a terrible shifting canvas. Declaring a match between a skin bruise and a suspect's teeth is highly subjective, which is why modern academic training is actively backing away from this to prioritize data-driven dental profiling. Now, let's leave the visible world behind and zoom down to the molecular level for our third pillar. This is known as touch DNA. Touch DNA targets the invisible skin cells that we naturally shed every single day just by interacting with our environment. Every time our skin brushes against an object, we leave behind thousands of invisible cellular traces. What makes this technique a game changer is its hypersensitivity. Modern laboratories don't need a massive bodily fluid sample, as they can just extract a full courtroom ready genetic profile from a tiny cluster of left behind cells. So you might wonder how this microscopic trace might actually solve crimes in the field. Based on the empirical work of Udianto and his team, everyday personal belongings like mobile phones and watches are excellent sources for touch DNA because they're handled constantly by their owners. The investigative process follows three core stages. First is swabbing, where the target object, like a cell phone, is carefully wiped down to gather these microscopic skin cells. Then it's brought into something known as CODIS analysis, where laboratories target highly specific regions across standard genetic locations to build a distinct profile. Finally, this leads directly to the match, and this provides a statistical identity check that is nearly 100% accurate when presented in a court of law. To go from that investigative swab to a data readout, the sample goes through a strict three-step molecular pipeline inside the lab. The first step is cell lysis and extraction. This is where the lab treats the swab with specialized enzymes such as proteinase K to break down cell membranes and isolate the raw DNA while washing away dirt and soils or oils. Second, because touch samples are so scarce, they undergo PCR amplification. The DNA is placed in a thermal cycler, and this acts like a molecular copy machine, and this replicates target markers millions of times in a few hours. Finally, capillary electrophoresis pushes the fragments through a microscopic tube using an electric current, and shorter fragments sprint through faster than longer ones, and a laser reads fluorescent tags to output a digital graph. When interpreting that graph, scientists are targeting something known as short tandem repeats, or abbreviated as STRs, and these are regions of our DNA that repeat in lengths that vary immensely between individuals. Following national forensic standards, labs look at specific locations called CODIS loci, and this focuses on markers like TH01, TPOX, and CSF1PO. By mapping the exact repeat count across all these standardized locations at the same time, the mathematical probabilities become astronomical. The odds of two unrelated people accidentally sharing the exact same multi-locus profile are literally billions to one. This is where the gold standard certainty of DNA evidence actually comes from. But science is only as good as the real world allows it to be, and not all touches leave behind the same quality of evidence. Pulling from the data by Ostage and Wernbach in 2017, biological recovery depends heavily on three environmental conditions. The first is surface type. Hard, smooth surfaces like glass and plastic yield a very high success rate because they preserve the cells cleanly. On the other end, metal objects like coins have a moderate to low success rate because certain metals oxidize and this breaks down the cells. The second is biology. Gripping an item with your non-dominant hand actually leaves behind a slightly higher skin yield due to increased friction, which is non-intuitive. It's easy to think that your dominant hand would actually have increased friction, like you would be able to grip something harder and leave behind a higher skin yield, but that's just not the case. The third variable is timing. On an ideal protected surface, these microscopic profiles can remain stable and retrievable for up to 40 days before they completely degrade. So, three variables: surface type, biology, and timing. This data proves that a touch DNA profile isn't a guarantee. It relies on these strict environmental and biological thresholds evaluated in studies shown by Ostage and Wormbach. An investigator must evaluate the background context of a piece of evidence immediately because, like I said in the previous slide, after 40 days, they'll completely degrade. Knowing whether an item sat outside on a porous surface or was heavily handled determines whether a sample is worth running through a costly molecular pipeline or if it's already lost due to degradation. If we bring all three pillars together onto a single matrix, the big picture becomes clear. Fingerprints are ideal for tracking presence at a scene via surface contacts, but the process is vulnerable to human cognitive bias. Forensic odontology offers unmatched biological durability for severe disaster scenarios, but become highly subjective when misapplied to bite marks on the skin. And touch DNA gives us extreme sensitivity for handling crimes involving weapons or daily items, but that sensitivity makes it fragile and highly vulnerable to secondary transfer. This means that your cells could end up on an item you've never even touched before. To conclude, true forensic identification isn't just about finding a trace at a crime scene. It requires an ongoing awareness of both hard science protocols and human psychological blind spots. From the visual checks of the ACE-V framework to the chemical steps of PCR amplification, the integrity of our justice system depends on holding human investigators to strict, objective standards. These advanced technologies don't exist to simply replace human judgment, they exist to inform it. Managing that intersection carefully is our ultimate path to a fair and a just legal system. These are my APA academic references. All of them are empirical articles and or studies. And thank you for watching. I hope you learned something.